The search for ways to harness the power of the immune system in the fight against cancer has led to the development of chimeric antigen receptors, or CARs. Kymriah, Tesogen Glycol Suspension for intravenous infusion, is the first U.S. Food and Drug Administration FDA-approved CAR T-cell therapy and has 2 indications. It is a CD19-directed genetically modified autologous T-cell immunotherapy indicated for the treatment of patients up to 25 years of age with B-cell precursor acute lymphoblastic leukemia, ALL, that is refractory or in second or later relapse, and adult patients with relapsed or refractory, RR, large B-cell lymphoma after 2 or more lines of systemic therapy, including diffuse large B-cell lymphoma, DLBCL, not otherwise specified, high-grade B-cell lymphoma, and DLBCL arising from follicular lymphoma. Limitation of use. Kymriah is not indicated for treatment of patients with primary central nervous system lymphoma. CARs are fusion proteins that can be specifically designed to target any antigen expressed on the surface of cells. First-generation CARs contain 3 domains: a targeting antigen-binding domain, a transmembrane domain, and an intracellular CD3 zeta signaling domain. This 3rd domain helps to couple antigen recognition with critical T-cell signal transduction pathways. Second-generation CARs added a co-stimulatory signaling domain designed to improve T-cell activation and persistence. Kymriah is a second-generation CAR containing a 4-1BB co-stimulatory domain. The co-stimulatory domain varies by CAR and plays a critical role in augmenting the CAR T-cell functions of expansion and endurance. 4-1BB has been shown to enhance early expansion and long-term endurance in vivo and in vitro, and induce central T-cell memory differentiation in vitro. It also may help Kymriah CAR T-cells evade native immunosuppressive stimuli in the body. Another co-stimulatory domain used in the creation of CARs is CD28. CD28 has been shown to be involved in early, rapid expansion in vitro and correlated with effector memory T-cell differentiation known to provide immediate protection with limited long-term persistence in vitro. Kymriah mechanism of action data are based on in vitro, in vivo data. The design of CARs determines several important properties. Most importantly, the antigen recognition domain enables CARs to target specific antigens independent of presentation by the major histocompatibility complex. Kymriah is designed to target the CD19 antigen. CD19 is a cell surface protein whose expression is restricted to B cells and their precursors, which are affected in B-cell leukemia and lymphoma. Kymriah is an individualized therapy in which CAR T-cells are created from a patient's own T-cells. Once a patient is identified as appropriate for Kymriah, they undergo leukapheresis at a designated treatment center. During leukapheresis, the patient's white blood cells are selectively removed from peripheral blood and the remainder of the blood is returned to the patient. The cells are then cryopreserved and shipped to the GMP-compliant manufacturing facility. Cryopreservation allows both the physician and the patient more flexibility in scheduling leukapheresis, and the shipping container provides a 1-week window to keep cryopreserved leukapheresis material frozen in case of weather-related delays. There, the cells are thawed and washed to remove cryopreservants and cellular debris. The leukocytes, including T cells, are isolated using centrifugation. These cells, including T cells, are then exposed to antibody-coated magnetic beads designed to promote proliferation of T cells and enhance the efficiency of their genetic reprogramming. After pre-activation, the T cells are transduced with a transgene that encodes the CD19-specific CAR protein. This is achieved using a lentiviral vector. Lentiviral vectors have been shown in vitro studies to have transduction efficiency because they can transduce both dividing and non-dividing cells. These genetic modifications reprogram the patient's T cells into Kymriah CAR T cells. Next, the cell population is expanded to produce enough cells for treatment. The Kymriah T cells undergo a rigorous quality control process. After the target cell count is reached and the cells are deemed viable, the reprogrammed T cells are cryopreserved and shipped back to the Kymriah Treatment Center for infusion. Patients are given lymphodepleting chemotherapy prior to receiving Kymriah. This chemotherapy is thought to facilitate CAR T-cell expansion and persistence in vivo. Once Kymriah is infused, these CAR T-cells may evade some of the body's native immunosuppressive stimuli. The Kymriah CAR can recognize and engage the CD19 antigen on the surface of B cells. This initiates a cascade of events that may induce target cell death. This also may release cytokines that promote expansion of the CAR T cells. After the initial B cell elimination, the 4-1BB co-stimulatory domain is thought to support CAR T cell endurance and immunosurveillance. A population of Kymriah CAR T cells may remain to potentially monitor and detect new CD19-expressing target cells. With these advances, Kymriah helps to usher in a new era of hope and potential transformation of the cancer treatment landscape.